The next speaker is Chris Karittevlis from the AAI Scientific Culture Service in Cyprus. And he's going to talk about linear and nonlinear connectivity measures and their validation through anatomy and recourse to raw signals. Okay, thank you, Chris. The stage is yours.
Thank you too. Hello, everyone. My name is Christodoulos Karittevlis. I'm in the first year of my PhD at AAISCS with my main supervisor, Professor Andreas Ioannidis, who is also the principal investigator of AAISCS. I'm an early stage researcher in the ICON network, which is a program funded by European Horizon to 2020 and Marie Curie. And today I'm going to give a talk about linear and nonlinear connectivity measures and their validation through anatomy and recourse to raw signals. But before starting my presentation, I would like to thank you very much for giving me the opportunity to participate and present in this very interesting and very insightful workshop. I really learned a lot from all of the presenters. So I would like to thank the organizers and all of the presenters, the researchers. So this work is a work that I was doing and I'm continue working on it with Michalis Papadopoulos and Shabam Tiwari, who are both also members, early stage researchers in the ICON network. Yes, as I said, this is an ongoing work of the last 3 months here at AAISCS between me, Michalis, and Shabam, and we are supervised by Professor Andreas Ioannidis. And a brief outline. Today, I'm going to explain and talk about a procedure that we follow to start analyzing MEG and EEG recordings and end up with some functional connectivity results that give us some information about the underlying sources in the brain. So in neuroscience, when we are talking about connectivity analysis using EEG and MEG data, there are basically two methods, two ways. We can do connectivity on sensor level, where we calculate the functional connectivity between the time series data recorded by different EEG and MEG sensors. So we have the EEG and MEG signals, and then we use some connectivity measures to calculate the connectivity in between pair of sensors. The second way is connectivity on source level. So we have the signals again, and then we apply some basic preprocessing. We solve the inverse problem, and we have now the time series data at the different regions of interest. And now the connectivity analysis can be applied between the different regions of interest. So what we did is somewhere in between those two methods. So we haven't done any source localization, but we are not only looking at the functional connectivity between EEG and between sensors. Instead, we are using some knowledge about the anatomy and about where we are expecting to find the sources, and also the basic principles of the generation of the signals, in order to say something about the sources by only treating and processing the EEG MEG signals. Before starting with the methods, I would like to give a brief background about the somatosensory system, since the data that we were working on are EEG MEG recordings of somatosensory stimuli. So in this diagram, we can see that when we have a tactile stimulation, so when we have a stimulation on our finger or on our median nerve of the wrist, which are both common type of stimuli, the information, the input is relayed through the nerve fibers, the spinal cord, and reaches firstly the ventral nuclei of the thalamus in the contralateral hemisphere of the stimuli, and then it also goes to the somatosensory cortex, S1 or S2. The somatosensory cortex can be seen here in this image. We know also from experiments with electroencephalography from human and also monkeys that the somatosensory cortex is activated around 20 milliseconds after the stimuli. And we do also know from literature that the thalamus is activated around 14 milliseconds after the stimulation. So let's continue now. The data that we used were provided to us by Mr. Marios Antonakakis. They collected this data with their collaborators in one study of 2019. And these data were also part of, they were given to us as a part of a course of MATLAB tools in neuroscience that we had here at the AISCS. So a brief description of the data. They had 5 right-handed subjects. The EEG recording was with 74 EEG channels. The MEG recording was made with 275 magnetometers. The stimulation was electric stimulation of the median nerve at the right wrist. So we had somatosensory stimuli. And the data sampling rate was 1,200 Hertz, and 1,200 trials were used. So what we did first, we applied some basic preprocessing to the MEG EEG data. So as I said before, this was simultaneous recording of EEG and MEG data. So we filtered the data in the frequency band of 20 and 250 Hertz. Then we removed the line noise using notch filter at 50 Hertz and its harmonics. Then we put the continuous preprocessed data into trials. So each trial was defined as with start point at 100 milliseconds before the stimuli and ends up to 200 milliseconds after the stimulus onset. We also removed some bad noisy channels, actually 7 EEG channels and 4 MEG channels. And then the final step was to re-reference the EEG data using the average reference. So let's continue with the analysis of the data. In this graph, we can see, as we call the butterfly plot. So this is the somatosensory evoked fields from the MEG data. This is the average signals of the 275 sensors across all the trials. Each color represents a signal for each sensor. And we can see a very prominent peak at 25 milliseconds. And we are expecting that this peak is related with the activation of the somatosensory cortex. The reason why it's 25 and not 20, as I said, with the T20 component, as I referred before, is because we had 5 milliseconds delay in the delivery of the stimuli. So the 25 milliseconds is actually the 20 milliseconds if we had the stimuli at 0. Now we can see the same plot, but for the EEG data, so the somatosensory evoked potentials. We can again see this peak at 25 milliseconds, which is a P20 component coming from the somatosensory cortex, S1. But what we also see is another peak at 19 milliseconds. And this must be related with a source coming from the thalamus, so the P14 component. The reason we can't see this peak in the MEG data is because the MEG, as we all know, is sensitive to the tangential component of the source, but the EEG is sensitive to both tangential and the radial component of the source. Then we proceed with some visual detection of the dipole patterns in the topo plots of the MEG data. Here we can see the topography of the MEG data, so all the sensors and the amplitude at time step 25 milliseconds. So this dipolar pattern here, we say that is related with a source from the source in the somatosensory cortex. And this is the same at the same time step, 25 milliseconds, but now for the EEG data. And we can see that these two topographies are vertically oriented to each other. And the reason for that is because of the generation of the magnetic field and the electric field. We know that the magnetic field is vertical to the electric field. So these two topo plots are, we are expecting that they are from a source, they are coming from a source in the Monocytosolic Cortex, there in the area 3B. Another interesting topography that we were able to see at 19 milliseconds. So the P14 component from the EEG data. So this topography here tells us that there might be a source somewhere in the center of the brain, maybe it's in the thalamus, that is radially oriented, and that's why we can see this high amplitude, positive amplitude in the center, and the negative amplitude around in the periphery of the head. So it's the way the current is spread in the head. If we have a source in the center, radially oriented. So as I said, there must be some correlation with the thalamus. So our trick that we use to continue with functional connectivity, before doing the functional connectivity, was to apply some spatial filtering on the EEG and MEG sensor level. So we select the sensors in this neighborhood of the region A with the high positive amplitude, and another neighborhood of sensors with low negative, very low negative amplitudes. And what we do is that we average the signals in each neighborhood, and we subtract the average from the signals in the area B from the area A. And this is what we get. So the blue line shows the average signal of the sensors in the region A. The green line now is the average of the signals of the sensors in the area B. When we subtract the two signals, we see that the signal from the underlying source contributed to the selected pattern of sensors is amplified. And also, any contribution from distant sources is eliminated, because if we have another source outside these two regions that is projecting its activity in the sensors in both areas, then by the subtraction, this source will be eliminated. So what we achieve by subtraction is that we focus more in the source that is exactly beneath the center of these two regions. This in the middle line, we take the center below this point. So we apply this technique for the EEG data at 25 milliseconds. And now we have this selection, and we have the reference signal, and we do also the same for the MEG at, again, 25 milliseconds. And we have now the comparison signal. This signal is from one trial. So then we apply the functional connectivity between those two signals that are both coming. We are expecting that they are both coming from the same source. We use the correlation coefficient. This is the map, is the average map of the correlation coefficients across 1,200 trials. And we see that there is something here at 50 milliseconds high correlation. And we also use the time delay mutual information. So the entropy of the reference signal plus the entropy of the convergent signal minus the combined entropy of the both signals with some time delay. Here we cannot see, we see that there is some high correlation, some high values of mutual information at around 50 milliseconds. But we need to work more on that to see if there are also other high values where we are expecting to be, because we believe that the high values here obscure the other events that might be happening, other correlation. So we need to work more on that. but the most interesting is what I'm going to show you next. So we applied the same thing, but now for between the source at 19 milliseconds, so the 14 milliseconds actually, the P14 component. Now, in order to select the sensors, we select an area in the center with the high positive amplitude, and the sensors in the outer ring here B with low negative amplitude. We use this spatial filtering to capture this radial source. So we have again the reference signal, which is the EEG, using this pattern here, and we want to compare it with the signal with the selection of the sensors here in the area A and B from the MEG data at 25 milliseconds. So the comparison signal now is the MEG at 25 milliseconds. So the reference signal is something coming from the thalamus. We're expecting something coming from the thalamus, and the comparison signal, we are expecting that is a signal coming from the Monocytosolic Cortex. And we want to see if there is any correlation between the two, any communication. So we again use the correlation coefficient. We are still not able to see something here using the correlation coefficient, but interestingly enough, we were able to see something in the mutual information map with the average of 200 trials. So we see that in the x-axis is the reference signal, so the EEG, the signal from the EEG at 19 milliseconds, and the y-axis is the time delayed MEG signal at 25 milliseconds. And we see that at around 10 milliseconds, there is the thalamus, so the source that the signal that might be related with the thalamus is sending some information 10 milliseconds after. So, and if we continue time, we see that this communication changes direction. Then the Monocytosolic cortex might be sending some information to the thalamus. So my point is that by only doing some spatial filtering to the sensors, and with what we know from literature and how the signals are generated, we are able to see that there is some communication back and forth between the thalamus and the Monocytosolic cortex. So our next step is to work on the source space. We want to use source localization algorithms to validate the location of the sources in the brain. We want also to solve the forward problem to generate EEG and MEG data from dipoles located at these assumed areas, for example, the S1 and the thalamus, and see how the Topo plots look like. And in this way, we can validate that what we use as spatial filtering is correct. And also want to do some source reconstruction, use some source reconstruction algorithms to extract the activation time courses of the sources. And if we have that, we will be able to validate our functional connectivity results from the raw signals by estimating the functional connectivity between the time course signals of the sources now. Yes, that would be all. Thank you very much. If you have any questions, please don't hesitate to ask me.
Thank you very much, Chris, for this very nice and interesting talk. Let me see if there are questions from the audience. I see a question from Annalisa.
Hi, Chris.
Hello. Thank you.
And okay, I read that in your last slide that a future step is to use some source reconstruction algorithm. So I'm wondering if you have idea which kind of algorithm, inverse algorithm you want to use.
Yes, I want to use the magnetic field tomography algorithm, which is a technique developed by my supervisor, Professor Andreas Ioannidis, which works very well on the MEG data and also EEG data. So, and we have all the software here. So yes, I'm going to use this. But I can also use some other algorithms from Filtrip to test and compare.
And also, sorry, did you record simultaneously MEG and EEG?
Yes, the experiment was simultaneous recording of EEG and MEG.
Okay, so you can reconstruct only from MEG data, from EEG
Yes.
and both.
Yes,
Okay.
that's the point, yes. To use both the information, the complementary information, and make some more results from this.
Okay, thanks a lot.
Thank you too.
Thank you. Let me check if there are other questions from the audience. If it seems not, so thank you again, Chris, for this beautiful talk, virtual
Thank
club.
you very much for giving me the opportunity.